Bismillah Rahman Rahim 9700 November 24 paper 22, and this is the second video, and we start off with question number 4. Now you can see the grade boundary for this is here, paper number 22. We're talking of paper number 22. So the total marks, maximum raw marks available were 60. Now 40 would get you an A, 33 would get you a B, and 27 would get you a C, and 22 would get you a D, and 16 would be a fail. No, 16 would be a pass, sorry. Less than 16 would be a fail, because E is a pass. So most of you ask me the grade, so 40 out of 60 is your aim. You must aim for something like 40 or more than 40, nothing less than 40. Starting with question number 4, adult stem cells are undifferentiated cells that are found in most animal tissues. Adult stem cells can divide by mitosis throughout their lifespan to form identical stem cells. Self-renewal, or to form cells that can differentiate into the functioning cells of that tissue. Mitosis is important for repair of tissues. Please do not write repair of cells. Cell cannot be repaired. And that's a very seldom we have an MCQ on that. And then, of course, you get it wrong because you think a cell can be repaired. No, cell cannot be repaired. It's repair of tissues. Explain what is meant by repair of tissues. Repair of tissues means replacing cells that are damaged. Replacing cells that are damaged or destroyed or worn out or old. So it is replacing cells, repair of tissues. So replacing cells that are damaged or worn out or old. Like for instance, you can say, oh, I'm replacing my pencil case because it's all torn, or the zip is broken, or something it fell down, and some of it is really got damaged. So replacing cells that are damaged, worn out, or old. Then the B part of the question says uncontrolled cell division is a characteristic feature of tumor formation from a differentiated cell. Describe other features of tumor formation from a fully differentiated cell. Number one, it is the result of mutation. Number two, it is got a faster cell cycle, or they're going to be faster cell divisions or continuous cell divisions. And there is no mitosis continues beyond the space available, so there's no contact inhibition. You see, when a cell comes in contact with another one, then its growth is inhibited. So there is no contact inhibition. That's a very technical word we use. A normal cell cycle checkpoints are not occurring. You know, usually a checkpoint occur, and then of course it goes on to the next stage. Loss of the original normal function. Cell cycle continues because there's no response to the cell signaling that okay, now stop mitosis. Cell do not carry out programmed cell death. So any two out of those. I'm not saying you need to write all of those. So result of mutation, shorter cell divisions, no contact inhibition, errors not checked, loss of normal function, no programmed cell death. You see, there's a programmed cell death. Cells have a normal lifespan and then they must die. Now these cells which are tumors, they don't die, they become immortal. So they keep on dividing, keep on dividing. Part C, telomeres prevent loss of genes. Adult stem cells have chromosomes with long telomeres. What are telomeres? Telomeres are the ends of chromosomes. So adult stem cells have chromosomes with long telomeres. Explain why long telomeres are an advantage to cells that carry out many cell cycles. Now that was simple, we know our understanding of the function of telomeres. In DNA replication, the telomeres shorten. So when DNA replication takes place and this chromosome forms another, then the telomere becomes shorter. Some of the telomere is lost. Ends of the chromosomes are protected. Telomeres do not contain genetic information, or you can say telomeres are non-coding. And nucleotides at the end of the DNA are lost. So Only 2 marks, and you have to give me all this information, not all of it, any two of it. So after DNA replication, DNA shortens. Sorry, telomere shorten is wrong. Telomere shorten. So after DNA replication, telomere shorten. Long telomere means replication can take place many more times. Ends of chromosomes are protected. Telomeres do not contain genes. Now we have a whole lot of information which we need to read very carefully and slowly. Hematopoietic stem cells, HSCs, are adult stem cells are located in the bone marrow of bones. HSCs have a role in the formation of blood cells. Figure 4.9 is an outline summary showing the formation of some of the different types of blood cells that can be formed from the hematopoietic stem cells. The first stage is the division of HSCs to produce progenitor cells. Progenitor cells. These cells are also able to divide by mitosis but are not stem cells. So the first time HSC divides into progenitor cells. HSCs remain HSC here, but CMP cells and CLP cells are progenitor cells. And then they become MEP, GMP, pre-B and pre-T. And then they become all these cells which we see now here: immature red blood cell, megakaryocyte, immature neutrophil, immature monocyte, immature B lymphocyte, immature T lymphocyte. And then they become a mature red blood cell. That's when they lose their nucleus. And the megakaryocytes become the platelets and the fragments of cells. Immature neutrophils become mature neutrophils, and the monocytes become something known as X. And the mature B lymphocytes results in Y. And the immature T lymphocytes become mature T lymphocytes, which are T helper and T killer cells. And now we have the question. With the reference to figure 4.2, explain why GMP cells, which are progenitor cells, cannot be described as hematopoietic stem cells. Differentiation has already started. There's no self-renewal. Cannot form all blood cell types. Only forms the immature neutrophils and the monocytes. So the GMP cells, this is the GMP cells. They're only going to form this and this. Why can they not be divided, called the HSC? Because the differentiation has already started. There's no self-renewal. GMP should become GMP if they're stem cells. Like here, what did you see here? HSC to HSC. This is self-renewal. There is no self-renewal here. GMP doesn't become GMP. GMP becomes immature neutrophil and immature monocytes. So this is differentiation has already started. And there's no self-renewal. They cannot produce a stem cell when they divide. Cannot form all the blood cell types. Only forms neutrophils and monocytes. Only forms neutrophils and monocytes. So the wording should be right. Differentiation has already started. No self-renewal. Cannot form all blood cell types. Only neutrophils and monocytes. Then we come to the part E of the question. Figure 4.1 shows that monocytes differentiate into cell type X, which has a similar function to neutrophils. Name cell type X. M for monocyte, M for macrophage. That's how I tell you to remember it. So you wrote macrophages and you got a mark for that. Then part F, cell type Y shown in figure 1 releases molecules with antigen binding sites. Name the molecules released by the cell type Y. Antibody. So you got a mark for that. Then we come on to the G part of the question. The differentiation of T lymphocyte begins in the bone marrow and continues in an organ known as the thymus to produce fully differentiated T helper and T killer cells. I hope you know where the thymus is. It's not in the neck, it's on the front of the sternum. Please do not mix it with the thyroid gland, which is in the neck. Thymus is on top of the sternum on your upper part of the chest, center of the chest. In the thymus, T lymphocytes that bind to self antigens are destroyed. So T lymphocytes that bind to self antigen, your own body cell antigen. Otherwise, the T lymphocyte will start destroying your own body cells, and that results in an autoimmune disease. Explain why T lymphocytes that bind to self antigen need to be destroyed in the thymus. Very simple. Next stage of the thymus release into, you see, this will then release it into the general circulation. And this will expose them to the self antigens on the body cells. This will result in harm or the destruction of the body cells. And we need to prevent this, otherwise this will result in formation of memory T cells. And we need to prevent because this will result in the forming of an autoimmune disease. And T killer cells will release substances to kill the cells. And faulty T lymphocytes then cannot bind to foreign antigens, or what we say non-self antigens. So after they have matured in the thymus, then they're released into the blood, and this will cause them to step up an immune response against the body cells, and this will of course kill the body cells. And also we have to prevent the formation of memory T cells. And if this keeps on happening, then this results in an autoimmune disease, and ultimately it will destroy the certain organs, and then of course the person is not going to live long after that. With this disease, he doesn't have a long lifespan. Now, coming to question number 5, malaria is an infectious disease caused by the Protoctus plasmodium. As part of its life cycle, Plasmodium infects human red blood cells. Researchers can compare hemoglobin from the red blood cells of a healthy person with hemoglobin from a person with malaria. Throughout the world, most deaths from malaria are caused by P. vivax and P. falciparum. Name one other species of Plasmodium that causes malaria. Well, you could have said ovale. That is the one that was left. It is written in your syllabus. And malaria E. So either ovale or malaria E, but the spelling must be correct. It must be correctly spelled. B part of the question: in the laboratory, oxygen at different partial pressures can be bubbled through a solution of hemoglobin to determine the percentage saturation of hemoglobin at each partial pressure. A graph constructed from the result is known as an oxygen dissociation curve. Figure 5.1 is an oxygen dissociation curve for normal adult hemoglobin in humans. Let's look at it. Now, we have the partial pressure of oxygen on the x-axis, and we have percentage saturation of hemoglobin with oxygen on the y-axis. So we have a graph like this. We've talked about this in the classes, and we've discussed this in great detail. I hope everybody is very much aware of this. Part 1 says, in the experiment used to obtain the results shown in figure 5.1, the temperature and pH were standardized. Explain what the researchers would consider when deciding which temperature and pH to use in this experiment. Naturally, body temperature. So body temperature use conditions occurring in the plasma or the blood of the humans. And the pH approximately could be 7.4 and the temperature 37 degrees Celsius. Oxygen uptake, hemoglobin activity will be affected by temperature and pH. So pH 7.4 and temperature 37 degrees Celsius would get you 1 mark. Approximately 7.4, pH 7 was also okay, and 36 or 37 degrees was also fine. pH of why? Because this is the pH of blood or the plasma. And the fact that the oxygen uptake would be affected by temperature and pH, because there's a lot of kinetic energy, the movement, diffusion of oxygen, and then of course all the proteins which are involved in this, and the oxyhemoglobin being formed. Then coming to part 2, using a different, more rapid technique, researchers compared the hemoglobin contained in red blood cells of a healthy person with the hemoglobin of a person with malaria who has been infected with P. vivax. By analyzing the results, the researchers concluded that the oxygen dissociation curve of a person with malaria would be shifted to the right. So let's look at this graph. When you look at this graph, it says that it will be shifted to the right. This is the right. With reference to figure 5.1, explain how a shift to the right of the oxygen dissociation would affect oxygen loading in the lungs and unloading in the respiring tissues in a person with malaria. Naturally, affinity of hemoglobin for oxygen decreases. Lower percentage saturation of hemoglobin with oxygen. More difficult to load oxygen in the lungs. Higher partial pressure of oxygen needed to reach the same percentage saturation of hemoglobin. And oxygen released more readily in the respiring tissues. So this is actually the Bohr shift, in which the affinity of hemoglobin and oxygen decreases and the oxygen is unloaded in the respiring tissues. But that we speak in the context of more carbon dioxide being produced. Now they have totally changed the question here, but they are actually checking whether you know that part of the syllabus as well or not. Affinity of hemoglobin for oxygen decreases, less oxygen binds to hemoglobin in the lungs, more difficult to load oxygen in the lungs, higher partial pressure of oxygen needed to reach same percentage saturation of hemoglobin, oxygen released more readily in the respiring tissues. Then we come to part C of the question. A red blood cell that is infected with plasmodium cannot carry out its function as effectively as a normal red blood cell. Describe how the size and structure of a red blood cell is related to its function other than the fact that it contains a very large number of hemoglobin molecules. So, number one, it's very small in diameter, so it's able to pass through the capillaries. A very small size, so just fits in a capillary, so they can all flow in a line. Then no nucleus and no mitochondria, so there is more space for hemoglobin. No mitochondria, so oxygen is not used within the cell. Otherwise, aerobic respiration would take place and pull up all the oxygen, and nothing would reach the body cells. It is flexible and can squeeze through capillaries. Then it's biconcave, and it's a biconcave disc, so it increases the surface area for the uptake of oxygen. And also the fact that it's biconcave, so it reduces the distance for the diffusion of oxygen to bind with hemoglobin. Because it's a biconcave disc like this, so the oxygen just has to cross in here, doesn't have to go to it, and just it binds with the hemoglobin inside this. So the hemoglobin is inside the red blood cells, it easily binds to it. So there were about 9 points, and you have to give me 4 only. So we have lots and lots of points, and that is why you have realized that when you do a lot of past papers, you have nearly covered every topic in some of the other paper. So you have really revised very well. But if you have done it with a lot of concentration and memorizing these mark schemes and these points, so the fact that if you wrote it was small size, so able to pass through capillary, I'm not going to give you a mark. The mark is for 6 to 8 micrometers, so able to pass through capillaries. But if you said small size, so fits in a capillary, then you got a mark for that. No nucleus and no mitochondria. Then you said no organelles. That's all 1 mark, because nucleus and mitochondria are all organelles. So no organelles was allowed. Though I don't agree with that. I don't agree with that. So more space for hemoglobin. No mitochondria, so oxygen is not used within the cells. Flexible, can squeeze through capillaries. Biconcave disc, so it increases the surface area to volume for the uptake of oxygen. Now coming to question number 6, the transfer of water from the soil solution to the xylem of the roots occurs by the apoplast and the symplast pathway. Apoplast along or through the cell wall, and symplast is cytoplasm to cytoplasm. Mineral ions can be transported dissolved in water. Describe the transport of water from the soil solution to the endodermis of the roots by the apoplast pathway, and explain why this pathway cannot continue at the endodermis. Now that's a very good question. Cell wall, that's the apoplast pathway, is, you can say, cell wall pathway. That will get you a mark. Movement is through the intercellular spaces, or you can say in the spaces between the cells. It's reject if you said intracellular. Intracellular means inside a cell. Like you have intracellular enzymes and extracellular enzymes. So intracellular space would be wrong. Intracellular enzymes are those which work inside the cell. So movement is through the intercellular. In between the cells there are spaces. So there it would be passed through that. Then the apoplast pathway reaches the endodermis and cannot cross because it stops with the Casparian strip, which is present in the endodermal cells. Then this is made up of suberin, which is a waxy, waterproof, impermeable layer. And then all the movement of water is down a water potential gradient. And this does not, this is the non-living pathway. It does not cross membranes until the endodermis. So all the points which I've just said, but then you need to understand this is a cell wall pathways, movement in spaces between the cells, epidermal cells in the epiplast pathway, or if you said the cells of the cortex, or you could say the parenchyma cells, then stops at the Casparian strip, and this is made up of suberin. Movement is down a water potential gradient, and this is the non-living pathway because it does not cross membranes until the endodermis. B part of the question, researchers investigated the mechanism of transport used for the uptake of potassium ions K plus into root epidermal cells at different concentration of potassium in the soil solution. Complete table 6.1 to provide information about the two different transport mechanisms that were identified by researchers. Now, against the concentration gradient. Well, of course, if it's against the concentration gradient, then we do need a membrane protein. ATP used, yes, and name of the transport is active transport. Active transport. Right? Down the concentration gradient, you need a membrane protein. Yes, because it's a potassium ion, so it will be facilitated diffusion. Potassium ions cannot enter through the hydrophobic part. So yes, no, ATP used, no, because it's facilitated diffusion from a higher to a lower concentration, and this is the name for it. So facilitated diffusion, and that would have got you your 3 marks. So what are the 3 marks for? 1 mark each correct column. So this column means this whole column is 1 mark. This whole column is 1 mark, and then this whole, because it's very basic. This is something that's more of an O-level question. So 1 mark for each correct column, and this got you your 3 marks, and it's a total of 7 marks. That finishes this paper. And thank you very much. And please leave a comment to let me know how you all are doing in the exams and what else can I do to help you all out.